almost 30 seconds, takes them almost 30 seconds more. And look, they had to decrease their force output or their muscle activation in the low oxygen condition. Now, how much of a difference do you think that 20 seconds means to performance? A lot. So think about this. You're standing at the corner. Okay, you're standing at the corner of, say, Pine, and a car drives by you at 45 kilometers an hour, which is, say, about the speed that they're going. Now, try to read their license plate as they go by, and then count 20 seconds. You couldn't even see what type of car it is at 20 seconds at 45 kilometers an hour having passed you. It would be that far away. That's the difference between 100th place in the world and first place. It doesn't seem like much, but a performance-based environment, it's massive. 1.5% difference in performance is considered meaningful between basically force, place, and medaling. 1.5%. And in this case, it was 3 times that. In terms of the improvement and deterioration in performance. Now, if I were to ask you a question, what does the muscle fatigue development look like? The force loss under these 3 oxygen conditions. So now, at the end of 5 kilometers, they take this cycle, they rip all the equipment off, they stick them on the bench, they stick that nerve stimulator in, and poof, they shock the leg. What would you think the level of fatigue development, the amount of fatigue or force loss would look like under the different conditions if you had to hypothesize? Would it be the same? Would there be more force loss in hypoxia compared to hyperoxia? Or would the loss of force be the same across the 3 different trials? So put your hand up if you think it would be the same across the 3 different trials. Put your hand up if you're not willing to commit to an answer. Okay. So, the amount of force loss across the different conditions was nearly identical. So at the end of 5 kilometers, these people are so finely tuned to understand their bodies, their muscles, their basically their ratings of perceived exertion, that they complete 5 kilometers when their muscles are completely exhausted to the same extent, even though in the 3 different conditions they maintain completely different muscle activation patterns and completely different power outputs. But at the end of the test, their muscles were pooched to the exact same extent. They lost the same amount of force. It's just that you lost that force with less performance when when oxygen delivery was compromised, and you lost that force with better performance when oxygen delivery was improved. So this concept of leaving it out all in the field is not bullshit. It's actually real. You can actually leave it all out in the field. 3 different exercise conditions, a standardized 5 kilometer task, and at the end they've exhausted their muscles to the exact same extent, even though their performance is statistically and meaningfully different. They push their muscles to the same point of fatigue after 5 kilometers, but they just did it with higher and lower performance in hyperoxia compared to hypoxia. So the magnitude of exercise-induced quadricep muscle fatigue at the end of the 5 kilometer time trial was not different across the conditions. Yeah.
Even though they all did it to the same point of fatigue, how do you know that that's their maximum fatigue? Like that's the maximum the muscles can go?
Yeah, you don't. I mean, if you took that person and then immediately at the end of 5 kilometer got them to do a Wingate, they would be able to generate more power output. And there's a whole debate about central and peripheral fatigue, but it's a very, very good point. But for the sake of this particular, example, for the sake of this particular experiment, is that for that physiological task of 5 kilometers, which is very relevant to performance-based, is that they were able to push themselves to the same level of fatigue despite the different oxygen delivery conditions. Which I think is, that's pretty cool experimentally if you see all of the Hogan stuff and the Hazler stuff and all those other studies that I tried to show, but not necessarily during a 5 kilometer time trial, which again, everybody should have some ability to relate to as a performance performance-based task. So if you look, again, now if you look at the mean integrated EMG activity averaged over the 5 kilometers, the mean power output averaged over the 5 kilometers, the time to completion, and the ratings of perceived exertion, you see exactly what you would expect based on the net drive and kind of O2 conformer hypotheses. Is that in hyperoxia, they were able to maintain a greater level of muscle activation, therefore supporting a higher power output, shortening the time needed to complete the 5 kilometers, but without greater ratings of perceived exertion. So, this idea of pacing and symptoms that are really important because they're telling you a lot. In high oxygen, you can, at a given symptom level, you can push the muscle harder for the same rating of perceived exertion and therefore perform better. Oxygen delivery is compromised, the same rating of perceived exertion translates into less muscle activation, less power output, and a longer duration of time to complete 5 kilometers. So during voluntary exercise, the effect of changing oxygen content on exercise performance is determined by the regulation of central motor output to the muscle. So again, the ATP demand conforms to the oxygen delivery. So here's the oxygen conformer concept. In order to prevent excessive peripheral muscle fatigue development. So, during this voluntary exercise, an individual will actually decrease their force output to try to make sure that their muscle doesn't get overtly and dramatically fatigued very early in exercise. It's just not sustainable. So they'll decrease their force output, decrease the activation of the muscle, conform to the oxygen state, and compromise their performance. So, to bring all this back, decrease in oxygen level. In this case, you would have the muscle would conform by decreasing motor drive and decreasing the force requirements without much perturbation in muscle metabolic state. Yep.
I have a question about this slide. So the first graph on the top left,
Yep.
it says that hypoxia has so less activity and fastest laterales. Is that what it means?
Yep.
But did we say that when you're in condition of hypoxia, you recruit more motor units?
That's in order to maintain a constant power output. But remember, this is during 5 kilometer time travel.
Oh,
So the muscle will conform to the oxygen state. It'll basically decrease its activity and decrease its power output when Sam is voluntarily controlling his own power output. If Sam comes to my lab and I put him on a bike and ask him to exercise for 314 watts, I'm telling him what to do. In hypoxia, he would then be forced to drive the muscle harder, so the EMG would be higher for a given power output. But here, because he can control, he's like, that doesn't feel good. De-recruit that muscle, decrease the power output so that he can maintain the same rating of perceived exertion, but at a compromised performance. So it's the key is appreciating the modality differences. I'm imposing the work rate on him, whereas he's imposing it on himself. So, in the setting of reduced oxygen delivery, an individual during a voluntary exercise will actually decrease the recruitment of their muscle and decrease their force output to try to maintain their level of symptoms and prevent excessive metabolic perturbations of the muscle. But this comes at the expense of exercise performance. You decrease your mean power output, you compromise your time to completion. If you increase oxygen delivery, you can maintain a higher motor drive and a higher force output without much in the way of greater perturbations at any given time, or any given, say, kilometer point, in the other metabolic drivers of oxidative phosphorylation, and also without much in the way of greater exertional symptoms. In this case, you'd have enhanced exercise performance. So be able to maintain a higher mean power output and complete 5 kilometers in a shorter period of time. So, coming back to the constant load stuff now. Because remember, that was the other part of this experiment that they did. They did a time to exhaustion test at 314 watts in the different oxygen conditions. And here, constant load cycle endurance time on the y-axis here in the 3 different conditions. So you can see, as I mentioned in the previous slide, if you had allowed those people, when they stopped them at 270 seconds, to keep on going, in normoxia they would have exercised longer than in hypoxia. In normoxia they would have exercised longer than in hypoxia, and in hyperoxia they would have exercised longer than in both normoxia and hypoxia. And indeed, that's exactly what happened. 314 watts. They do it for 270 seconds, then they do it for 500 seconds, and then they do it for almost 1200 seconds. But despite those dramatic differences in exercise endurance time, when they strip them off the bike when they can't sustain 314 watts anymore, look, the muscle is equally as fatigued, it lost as much force. So they did almost 6 times more work in hyperoxia, but despite that, they lost as much force as a consequence of exercise when oxygen levels were high compared to when they were low. So that's the impact of how much you can shallow out that rate of development of fatigue. How basically how slowly you can make those fatigue-causing metabolites to accumulate. But I think what's important here, like I said before, remember the Hogan studies during the calf plantar flexion exercise? That despite the differences in power output and time, is that the inorganic phosphate levels and phosphocreatine levels were similar across the different conditions. That's really how you should interpret that twitch force. It took longer for them to fatigue, but when they fatigued, the muscle wasn't able to support that given power output any longer. They reached that performance limit. Oxygen levels just dictated the time that was required to do that. So at a constant power output during exercise, changes in oxygen content affects at exercise time to exhaustion. Hypoxia was less than in normoxia, and normoxia was less than hyperoxia. This conforms to the net drive hypothesis. Differences in oxygen content, however, had no effect on peripheral locomotor muscle fatigue at the end of exercise, suggesting that the muscle metabolic state was independent of intracellular PO2 at end exercise. So the rate at which the metabolites accumulated was dependent on oxygen levels. But at the point of exhaustion, they're independent of oxygen levels because presumably they're similar. You've degraded phosphocreatine to a similar extent, accumulated inorganic phosphate to a similar extent, generated or produced as much lactic acid to a similar extent, it just took you longer to do it. But at peak, the muscle metabolic state is the same and therefore independent of the oxygen conditions. The time required to achieve this critical metabolic state, however, does depend or is dependent on the intracellular PO2, which is evidenced by differences in cycling time to exhaustion. Is anybody monumentally confused, or does it all connect? Yeah? So, to try to bring this back to this concept again. In hypoxia, you exercise for 270 seconds. In normoxia, you exercise for about 500 seconds. And in hyperoxia, you exercise for about 1200 seconds. The rate of fatigue development, which is a direct reflection of the rate of phosphocreatine degradation and inorganic phosphate accumulation, is accelerated in hypoxia, attenuated in hyperoxia, such that your exercise endurance time will vary with the oxygen state of the cell. But at the point of exhaustion, the metabolic state of the muscle is very, very similar. So it's like the example. If we have to all travel to Ottawa, same distance, right? 215 kilometers. If I'm driving a Lambo, somebody's driving a Hyundai, I'm going to get there faster, right? You cover the same distance, but it takes you longer. So in the end of it all, you can get there you can basically, you have much better performance when your muscle is kind of receiving gas like a Lambo than it is a Hyundai. You can complete that task for a longer period of time. Performance is enhanced. So the net drive hypothesis, again, to beat a dead horse, at a constant, at a given ATP demand and a constant force output, if you decrease intracellular oxygen levels, you have to recruit the muscle to a greater extent to support that force output. But in recruiting the muscle, you're recruiting glycolytic fibers, these bigger and faster motor units. That's going to increase the rate of glycolytic flux. It's going to degrade phosphocreatine, result in an accumulation of inorganic phosphate. It's going to stimulate glycolysis, impair muscle metabolic function, and contribute to an exaggeration of exertional symptoms. Compromising exercise performance. So shortening the time to exhaustion or decreasing the power output that can be achieved. Alternatively, at a constant motor drive, at a given force output, if you can increase oxygen delivery, say by increasing oxygen content or increasing muscle blood flow, you can get away with recruiting the muscle less aggressively, so decreasing the motor drive to that muscle. You would have less in the way of exertional symptoms, so ratings of perceived exertion would be lower, and you wouldn't need as much of a change in the other drivers of oxidative phosphorylation, glycolytic flux, phosphocreatine degradation, to support that matching of ATP supply and demand. And under these circumstances, exercise performance would be improved. You'd be able to achieve a higher power output during an incremental exercise test, or you'd be able to exercise at a given power output for a longer period of time during a constant load exercise test. Okay? So, at the beginning of next class, what we'll do is that you'll all kind of get, I'll have printouts for everybody, and we're going to go through some examples where you're going to have basically all of these different parameters. So intracellular PO2, VO2, ATP, force output, all these different things, and you're going to have to draw the time course of changes from baseline to the one stage of exercise, a manipulation of oxygen, another stage of exercise, a manipulation of oxygen, another stage of exercise, to show that you have an appreciation for how, in response to manipulations and oxygen delivery, all of these different parameters would change if we were conforming to the net drive hypothesis or the oxygen conformer hypothesis. Okay, so you kind of, and those examples will be really critically important for ultimately what's going to come on kind of the working example of the final exam. Okay? And then from there, we move into pulmonary, which again, the lungs are the mediator, a major determinant of oxygen delivery. So we need to appreciate how the lungs exchange oxygen, because they're a pathway to make sure that oxygen gets to the muscle. And you should hopefully have a pretty good appreciation for how important oxygenation of the muscle is now to exercise performance. Okay? See you on Thursday.